What we're going to talk about is ethylene oxide sterilization. My name is Floyd. I've been at Nelson Laboratories for about the last 19 years doing ethylene oxide validation. So it's something that we've gone through and taught a lot of people how to do this. Before we talk about the validations, though, we want to talk a little bit about ethylene oxide in general. It's important to understand the history about how it works. When you understand how it works as a sterilant, it will help you understand if you're designing product or packaging to make it where you'll be able to successfully sterilize it. So we want to talk a little bit about the history, a couple definitions that are key that differentiate ethylene oxide from things like radiation. We're going to talk about the testing that you have to do up front, and then also the validation itself when you're at the time where you have your load designed and you're ready to sterilize at the contract sterilizer. And then there's a whole list of standards that are international and national standards that have to be followed. So if you look at the history of it, it was first discovered back in 1859 by a chemist named Charles Wurtz. It was one of those things where once they discovered it, they didn't really use it for sterilization. It was probably some 70 years later before they started using it for, where they decided that they determined that they could use it for insecticidal properties. And then in 1933, the first patent was designed for using ethylene oxide to sterilize or to destroy microorganisms, which is closer to what we use it for today with medical device sterilization. And then it was the late 1940s before it was first used for medical supplies and instruments. And that process really hasn't changed much then to what it is today. There have been some technical advancements, but the main process is fairly similar. And why we bring these dates up is the very important thing about that is because ethylene oxide has such a long history of successful use, it gets really gains wide regulatory acceptance. So the FDA is very familiar with it. If you're going to use ethylene oxide versus some newer technology like hydrogen peroxide vapor or something like that, you're going to have probably a little bit of a harder time than you would with something that is established like ethylene oxide. So if you look at the properties of it, what makes it such a great sterilant is that it's so reactive. It's always trying to open up and react with other chemicals. It's a colorless, odorless gas, so you have to remember that it's a gas, and when you're designing your packaging and your device, you have to realize that this gas has to be able to get into the medical device, and the hardest to sterilize reaches of that in order to get in there. It does have strained bond angles, so it's always trying to open up that epoxide ring, and it's very versatile as a chemical intermediate. So it's just a small percentage is used for medical device sterilization. It's mostly used industrially to make things like ethylene glycol and other things. So just to give you some facts about that, just in 2009 there was over 42 billion pounds of ethylene oxide produced annually, so just a huge amount. 99% of it's used as these intermediates that I just mentioned. 60% used for antifreeze, and then it's also used for polyesters, detergents, brake fluids, things like that, so a lot of other chemicals. But only less than 1% is used for medical device applications. So that's something to keep in mind. For what we do, it's the only thing we use it for, but that's just a small, tiny percent of what we do use it for. It is toxic, which is one of the things that makes it a great sterilant. So OSHA has set limits on the worker exposure. If you're going to be around it, you have to be cognizant of these levels. It has a permissible exposure limit of 1 part per million, which is pretty low, but it is a toxic compound, so you do have to take precautions and do monitoring if you're working with ethylene oxide. But because it has been so widely used, it's probably one of the most widely studied chemicals out there. And so there's more knowledge about it than most other chemicals that are. So that's something to keep in mind about the toxicity of it. It is flammable and explosive. 100% ethylene oxide is explosive. And the interesting thing about ethylene oxide is its range of flammability is 3% all the way up to 100%. It can create its own oxygen as it burns. So you have to, when you compare that to propane, it's only going to be flammable up to around 9.5%. So if you had 100% propane, you're not going to be able to burn that because it doesn't have a source of oxygen. But ethylene oxide is flammable through a wide range of that. So you have to be careful with it, not only for its toxicity but for its flammability. Static electricity can set that off, as well. It is as you can see in this picture, a sterilizer that had an issue, an explosion with that. It can be a very violent reaction if it's not, not the necessary safety precautions are taking place. If you look at ethylene oxide, there's 4 parameters that determine if you're able to sterilize effectively with that. This is one of the more complex types of sterilization. If you're looking at radiation, you just have 1 parameter, that's just the dose. Or if you look at steam sterilization, you have 2 parameters, you have temperature and time. But with ethylene oxide, you have 4 separate variables. And each one of these has to be working in conjunction with the other to have a successful cycle. So it's very closely monitored. You have temperature, relative humidity, gas concentration, and then obviously your exposure time, or how long it's going to sit in the chamber to sterilize. So temperature is one of the biggest items that we look at for people that are going to, if they have an issue with their cycle where they're having positives when they're first trying to design a sterilization cycle. The first thing we ask them is how high can you go up in temperature? Typical cycles are going to end up being around 120 to 130 degrees Fahrenheit, so about 55 degrees Celsius. You really don't see too many cycles that go much above 55 because it's mostly used for devices that are heat sensitive. Things that can't be sterilized with steam because the plastics would melt. They can't be sterilized with radiation because a lot of the polymers will degrade, or they'll crack or cross-link. So there's a lot of material compatibility issues with radiation. So ethylene oxide's real niche is the low temperature type of sterilization for those. The increase in temperature may also increase permeability. It may help the gas penetrate in. Remember, it's a gas, so the molecule has to penetrate in through the packaging and through the device and all the different hard to sterilize locations. Relative humidity is also needed. It's critical for the reaction, the alkylation reaction to take place. So it almost acts as a carrier to help pull the gas into the product, into the packaging. The optimum humidity level is typically 35% at the spore level, but you'll see most industrial sterilization cycles be around 65 to 70% relative humidity. That's pretty standard. It's also going to help transfer, like we mentioned, it helps transfer the EO through different materials. If you look at something like nylon, if you have dry ethylene oxide, it's not going to penetrate very readily, but if you add moisture, then it'll readily penetrate through nylon. So the moisture is definitely critical, which sometimes presents a challenge if you have absorbable sutures or other things that are moisture sensitive, but it works real well that way. Gas concentration, that's basically how much gas you are putting in the chamber. You're going to add the gas up to a certain target point. Normally that's expressed in terms of milligrams per liter. Typical industrial sterilization cycles you'll see are 400 to 650 milligrams per liter. That's pretty typical. If you look at something like vaporized hydrogen peroxide, their gas concentration in a typical Sterad or Steris VHP cycle is going to be about 1.2 milligrams per liter. So if you looked at it, compare that, it would seem that peroxide would be a lot better of a sterilant, but the problem with peroxide is it does not penetrate. The real advantage of ethylene oxide is it can penetrate into most plastics. It's amazing of what it can get into given enough time. The things that you will not be able to sterilize with ethylene oxide are things like glass ampules. Sometimes you'll see lidocaine vials that they'll put inside a kit. You're just going to sterilize the outside surface of that, so it won't sterilize liquids or glass ampules, or if you had foil pouches, you have to have some type of breathable material like Tyvek. Tyvek or paper pouches are pretty standard, or trays with Tyvek lids are pretty much the norm for packaging for ethylene oxide sterilization. It is a gas again, so it has to be breathable. So then you look at your exposure time. That's how long it's basically just sitting in the chamber. Once you reach that target pressure of gas, this is where the sterilization is taking place. And this really depends on how hard the individual product is, but a normal time is going to be anywhere from 2 to 10 hours. Just depending on that. So the exposure time, again, once it gets to where it needs to go, it's going to sterilize very rapidly. The problem is sometimes you have to penetrate through multiple layers of packaging, through the actual plastics of the device into the center of the device. So once it gets there, it's going to sterilize very rapidly. But then once you're completed with that, then you're going to also have to remove the ethylene oxide since it is toxic. So a lot of times the harder it is to get into a device, the harder it may be to get it back out at the end of the cycle. So that's important as well. When you're looking at ethylene oxide, you have to realize that it's an overkill process. So you're not setting your cycle length based on your bioburden like you are with radiation sterilization. You're using a biological indicator in what's called an overkill cycle. And what they're going to do is they're going to use biological indicators, which are basically, most people have seen these paper strips. They're basically just a piece of filter paper that they put spores of an organism called Bacillus atrophius. That's the indicator organism for ethylene oxide. And there's lots of, your biological indicator can be pretty much anything that you put those spores on. It may even be your actual device itself. It may be a piece of surgical suture that you inoculate. You have to have 1 million of these spores in the hardest to sterilize location, and you have to be able to kill that in half the time. So you can see why they call it an overkill process, because you're killing these highly resistant spores, and spores are like a seed is to a plant, where it's going to be very highly resistant to heat and chemical attack. So you'll put these spores in the hardest location. The bioburden on the device may only be something, you may have a bioburden of less than 10 organisms on your device, but you're going to monitor that with 1 million of these very, very resistant spores, and then have to show that you can kill that in half the time. So you can see why it's called an overkill process. So the biological indicators are very important. In order so you don't have to use your actual product, if your actual device is very expensive, maybe a catheter that costs $1,200 each, you wouldn't want to have to use those every time you do a sterilization cycle to place these biological indicators in. One cycle, you might need to have 20 of these biological indicators placed throughout the product load, so you wouldn't want to have to spend $20,000 of product in order just to waste to use these biological indicators in there. So a lot of times we come up with what's called process challenge devices, or a PCD. And what these are something that's going to simulate the resistance of the device. It doesn't have to look like the device, it just has to have the same resistance to sterilization as the device. So it may be something that's cheap and easy to work with, a syringe with some type of cap or tubing, something that the gas has a hard time getting into, so that we can make those biological indicators as hard to sterilize as maybe the internal lumen of a catheter with only a few thousandths of an inch clearance. So there's a lot of variety of different PCDs, and you really have to choose one that has the same resistance as your product. So some of the initial testing that you have to go through, you go through all of this stuff up front before you're going to do your validation. You even have to think about your biocompatibility testing. You want to do your biocompatibility testing on samples that have been exposed to the sterilization process. That's true whether you're using radiation, ethylene oxide, steam. A lot of people don't do that. They'll do their biocompatibility testing but not do it on sterilized product, and that's something that sometimes can cause issues with some of the testing. So you want to do your biocompatibility. Obviously your product functionality, you need to make sure that when you use ethylene oxide, the device isn't going to fall apart, the adhesives aren't going to fail, whatever packaging you have is not going to burst because of all the pressure and vacuum changes that go on in the cycle. You're going to do your package testing as well. It's got to be breathable. You want to do comparative resistance testing. What that is, we'll go into this in a second. This is kind of the cornerstone of the validation, and that's going to be where we determine how hard the product is to sterilize to help set the cycle length. And then there's the bioburden resistance. Because we're testing with these biological indicators, we're going to release the cycle if these BIs are killed, but we haven't tested the actual product. So at some point you have to tie those two together to show that if the BI is sterilized, that there is no organisms on your bioburden that are more resistant than that BI organism. And then you're also going to have your population verification of your spore strips. You have to show that there are 1 million spores per location, a minimum of that. So the first one is this comparative resistance test. It's going to serve really two different purposes. You're going to determine how long it takes to sterilize the product, where on that product is the hardest location, and then you're going to find that we want to come up with some type of PCD that we can use to monitor the cycle so we don't have to use the actual product. So we're going to use, because we don't know which PCD is going to be most appropriate for the product, we're going to use a shotgun approach basically, and we'll try 4 or 5 of different varying resistances and do them side by side with the product, and then use the one that's most appropriate to the actual device. So what you're going to do is basically, in the first picture here, you inoculate your devices in all the different locations that may be hard to sterilize. You're going to put these BIs, they have to be in the hardest spot to sterilize, so you got to make sure you inoculate everything that may be difficult. And then you're going to run fractional sterilization cycles. So you're going to take a group of samples, usually 5 to 10, that you may do, 30-minute cycle, maybe all the products and the BIs grow, so you're going to increase the time, go to 60, 90, 120, whatever time you do, and then as the easy sites of the product start dying off, and then you're left with that most resistant spot on the product, and you're able to find out, first of all, what is the most resistant location, and then how long does it take to sterilize that. And then that's basically what your half cycle time is going to be, because if you remember, your half cycle time is the time that it takes to, you have to show, that's the time that it takes to get complete kill, and then you're going to have to double that for your routine cycle. So the half cycle for your validation will be this point where you get complete kill of all the different locations. And then you're just going to do basically a sterility test. You've inoculated those products in all those locations. You're going to pull those, whether they're spore strips or surgical suture that's inoculated with spores, or maybe you had to inoculate the device directly. You're going to take all those locations out, test them in microbiological media, and incubate those for 7 days. And you're just looking for growth or no growth. So you're looking at a comparison between the two. Where's the point where the product's killed off? Where's the PCD that's most appropriate for that? So that's the comparative resistance test. Bioburden enumeration, most people are familiar with bioburden. What bioburden is basically the number of organisms that are on the device just through your manufacturing process. You know, bioburden is going to be just from people handling the device. There's going to be bacteria, mold, yeast on people's hands as they handle materials. The raw materials are going to have bioburden on it. You're going to have organisms just settling out of the air, landing on the device. So all that's bioburden that has to be sterilized and killed. And so it's important to know how many of those are on there. So there's two different types of bioburden testing. The first one is bioburden enumeration, which is going to tell you how many organisms are on there. And that's a test that you typically do quarterly. And it's a judge of your manufacturing process of how clean the device is over time. As you add new people to your clean room staff, your bioburden may tend to go up. So it'll start, it'll kind of go up and down over the year, but as you monitor it over time, you're going to see trends in that. So it's important to see how that goes. But with ethylene oxide, you're not setting the cycle based on what your bioburden numbers are like you are with radiation. In radiation, if your bioburden was, say, 269 organisms, there's a table that you would look at. Go down to 269, you'd find what dose you have to dose the product at. With ethylene oxide, we're using this overkill approach, so the numbers aren't quite as important as the resistance of it. And so the bioburden resistance, that's how hard they are to sterilize. That's more important than the numbers. Sometimes you can see you might have a device that has 10,000 organisms on there that's very easy to sterilize because these organisms are not spore formers, they're not resistant. So the resistance is something that is a second part of that you have to show. And basically you're going to do that the same way as you did the comparative resistance test. But instead of us inoculating the device with these spores, we're just going to take the device as is, place it in the sterilizer with these process challenge devices that we came up with, and show that PCD is more resistant than whatever natural bioburden is on the product. So it's two similar tests, but they're accomplishing two purposes. So again, you're going to use non-inoculated product, equal numbers, so you may take 10 samples of your product, 10 of whatever PCD was most appropriate, run these fractional cycles again, and then test for sterility. So again, you want to find the point where in this case, you try to find a point where the BIs are positive, because you want your BIs to grow, but the product to be killed off. And that way you can clearly show that BI is the most resistant entity, and that BI is appropriate to monitor your sterilization cycle. So once you've done all this upfront testing, you're basically down to the validation process. And a normal validation will consist of an empty chamber profile, which is basically repeated once a year. That's to make sure that the sterilizer is still operating correctly, that it's within the same tolerances. There's a requirement that the empty chamber profile during exposure, during that sterilization part of the cycle, has to be a range of plus or minus 3 degrees Celsius. So there can't be a bigger range than that for the empty chamber. And then you're going to do a fractional cycle, again, just to show recovery of the organisms, that if anything were to grow, that you would be able to detect it in whatever sterility test you're doing. And then you're going to do 3 half cycles. The half cycles are where you're going to prove the sterility assurance level. That's where you're proving that the product is sterile. Because remember, you have to kill it in half the time, and then your routine cycle is going to be double that, and that's called a full cycle. And then you're going to have to do full cycles to do the EO residual testing. Because that product's going to go through that full uninterrupted cycle, you're going to have to test samples that go through that as well. So it's basically one of the things where you have to repeat everything in triplicate just to show repeatability. And just like many validation processes. So in those validation cycles, what you're going to have is you're going to have biological indicators that are spread throughout the load. In a lot of cases, these are multiple pallets of product, and you're going to seed the load with these different process challenge devices that we determined in that comparative resistance test. Once the cycle is complete, they're going to pull those out, test them, and that'll show if the load was adequately processed or not. So the numbers of biological indicators that you're going to use for any specific process is based on your load volume. So how big the load is, whether you have 1 pallet or 2 pallets or 3 pallets. It used to be based on the chamber volume. So if you were sterilizing a shoe box in a 7-pallet sterilizer, you had to have enough BIs and temperature probes as if you had filled that whole thing. So that was kind of a strange quirk in the standard that they've just changed now. Now you just have to have the number of BIs and temperature probes based on the actual amount of product that they do. There's a formula in the standard that goes through that, and it'll tell you exactly how many to use. You're going to also have temperature and humidity probes in these. And again, remember, temperature and humidity are 2 of the main parameters for ethylene oxide sterilization, so you have to carefully monitor those through your validation cycles and set your own ranges and tolerances for those. Then you're going to have your residual testing. Again, ethylene oxide is toxic, so you need to remove it from the product. There's specific limits for a device. For most devices, they've gone down from 20 milligrams to ethylene oxide down to 4 milligrams. So the limits have been reduced of what's allowed on the product. So you have to go through some testing where it extracts all the ethylene oxide out of the device, and they quantify that to see if you meet those limits before they can be used on patients. And the limits are based on the patient contact of the device. So if it's an implant, it's going to have one requirement. If it's just a device that's used for less than 24 hours, it's going to have a separate requirement. And basically, this is the new results. So if it's a limited exposure device, something that's used up to 24 hours, you have 4 milligrams of ethylene oxide and 9 milligrams of ethylene chlorohydrin. And then the second category is prolonged exposure devices. And these are basically the same 4 and 9 that you have in the first 24 hours, but then they add a caveat that you can't exceed 60 milligrams in the first 30 days. And then finally, the last category then is going to be an implant, something that's going to be in the body forever. You basically have to do what's called an exhaustive extraction. That's where they're going to keep extracting it until they get all of the ethylene oxide out of the device, and then they keep the same limits that they had before. So you have the 4 for the first 24 hours, the 60 milligrams for the first 30 days, but then they add this 2.5 grams in a lifetime. So that's the ethylene oxide, and this is a lot of text. I don't expect you probably necessarily to read it all, but if you have any questions about that, let me know. But it's really just based on the classification of the device and how long it's going to be used on the device. So then once you've validated your process, you've gone through all those cycles, the empty chamber profile, you've gone through 3 half cycles, 3 full cycles. At some point then you have to revalidate that process. Now this is where a real advantage of ethylene oxide is. With radiation you have to do quarterly validation. So every quarter you're going to be revalidating that cycle because it's so heavily based on your bioburden. But with ethylene oxide, what the standard states is that you look at your original validation approximately every year or at defined intervals, and then you decide to what extent you need to validate. There's only a requirement to do an actual physical half cycle every 2 years, and that's found in AMI TIR 16. So that's one big advantage. Usually the initial EO validation is going to be very costly, so it's going to be, a lot of people are shocked by the cost up front. But over time, you're going to recapture that back because you may only be doing a revalidation cycle every 2 years as opposed to every quarter or even every year. But the current industry standard, that being said, the standard states that you can do it every 2 years. The problem is FDA still pretty much expects people to do it once a year. So normally the revalidation is going to consist of just a single half cycle, and that's just to really confirm that sterility assurance level is still being met. So just one half cycle once a year or once every 2 years is what the revalidation process consists of. So here's a big list of standards, and I can email this list to people if you want to contact me after this or get one of my cards. These are all the standards that are required for an ethylene oxide validation. The first 2 are the most important. That's ISO 11135. It's basically a 2-part standard, and that's the most critical one. That's where all the requirements for the validation are found. There's a lot of other standards that help you meet this, or they give you guidance on how you're going to meet ISO 11135, but that is the main standard that you have to meet worldwide. ISO 10993-7, that's the EO residual guideline. That's where all those limits we talked about are found, and that's ISO 10993-7. And then there's a lot of these AMI, what they call TIRs, or Technical Information Reports. These are ones that that we as Americans, the American Association, the Association for the Advancement of Medical Instrumentation, they write these to give guidance on how to meet the standards. So they're more guidance documents as opposed to actual standards. But put an asterisk or write down TIR 16. That's probably the most valuable one that gives you the most guidance on how you do the validation process itself. It gives you, if you have different sizes of loads and all kinds of different things, it talks about that. There's also AMI TIR 28 that talks about product adoption. What that is if you have a currently validated cycle that you've already validated, but you have a new product that comes down the line, it allows you to adopt it into your current cycle without totally going through the validation process again. You just have to compete the comparative resistance test to show that the new product still is under the resistance of that process challenge device that we determined initially. So these are basically all the standards for EO. There's 11138. These are the biological indicator standards. If you're using commercially made biological indicators, you don't have to worry about that because they all have to comply with these standards. So there's not a lot you have to do there. ISO 11737 is part 1 is the bioburden standard. So it talks about doing the bioburden testing. Part 2 is going to be the product sterility testing. So that's it. That's all the different standards that you have to meet. As you can see, there's a lot of different requirements for that. So this was a real brief, kind of high-level overview. We typically will do this ethylene oxide validation talk in about 4.5 hours. So you can see we tried to shrink it down into this time period. So if there's any questions, feel free to. I'll just be out here afterwards if you have any questions, and I'd be glad to answer any questions that you have. But we thank you for your time, and thanks for coming, and enjoy your day at the show today. Thanks.